Bonjour tout le monde, bienvenue à cette conférence de yeast. Aujourd'hui, je suis content d'accueillir Greg Lang comme conférencier. C'est la première fois que j'ai entendu parler de Greg. On était dans le même building, j'ai une de mes collègues de laboratoire qui était allé à un cours gratuit, dans lequel Greg était. Il est revenu au laboratoire, il venait avoir un cours, il y avait un gars qui était tellement brillant, il venait de toutes les étapes de la méiose, de la mitose. Il racontait ça au professeur, la personne dont on parlait, évidemment, c'était Greg. Donc, nous, on avait étudié ça depuis le bac, lui, il se souvenait des détails de la vie. Donc, Greg a fait son doctorat à l'Université Harvard. Il était dans le laboratoire de Harry. Il a fait beaucoup de travaux fondamentaux sur la régulation du cycle de l'homme. Il s'est intéressé tard dans sa carrière à l'évolution. Et c'est là que Greg a commencé à travailler avec Enzo. Et on n'a pas beaucoup interagi quand on était au doctorat parce que c'est dans le laboratoire, notre laboratoire, peut-être un petit peu en compétition, ce qui est dommage parce que si on s'était connus plus à ce moment-là, je pense que ça aurait été une bonne occasion de travailler ensemble. Et Greg, à ce moment-là, pendant ses études doctorales, a fait une des expériences les plus brillantes que j'ai vues dans ma carrière, c'est qu'il essayait de voir l'effet de la position des gènes sur leur taux de mutation. Donc, ce qu'il a fait pendant son doctorat, c'est de prendre un gène chez la levure, de changer de place dans le génome, de mettre un peu partout dans le génome, mesurer le taux de mutation dans ce gène-là pour voir comment ça variait le long des chromosomes. Ça, c'est avant qu'on puisse séquencer des centaines de génomes, donc il fallait être un peu plus inventif pour maintenir des expériences comme ça. Donc, Greg a fini son doctorat en 2007 et est parti ensuite à l'Université de Princeton. Il a fait un postdoc avec David Hotstein, qui est un des fondateurs du domaine de la physique et du développement des outils en évolution. Pas tellement en évolution, dans ce cas-là, c'est plus en génétique classique des levures. Et là, il a continué à travailler sur différents aspects d'évolution. Entre autres, il a fait des expériences d'évolution expérimentale en utilisant des techniques de cryptage dans le débit, dont on va parler aujourd'hui. Et après son postdoc, il a eu un poste à l'AI University en Pennsylvanie, où il est depuis 2013. Il est présentement professeur, professeur agrégé, et il est aussi directeur des études graduées là-bas, donc il connaît aussi bien les programmes de formation. Et l'autre chose importante qu'il fait, c'est qu'il est instructeur au cours de génétique et là-bas à Côte-du-Spring-en-Bas. Donc, à chaque été, Côte-du-Spring-en-Bas organise un cours pour former des scientifiques à travers le monde qui s'intéressent à la génétique de l'eau et qui n'ont pas de formation nécessairement en génétique de l'eau. Donc, ils envoient une quinzaine de personnes à Côte-du-Spring-en-Bas. Ils passent quelques semaines ensemble, ils font des expériences presque 24 heures sur 24. Et Greg est un des instructeurs à ce cours-là. Donc, il aide vraiment beaucoup à développer l'expertise et à répandre les connaissances qu'on a sur la génétique de l'eau. J'ai eu la chance de visiter l'été passé. Et c'est vraiment impressionnant le type de formation dans si peu de temps. Quand les étudiants partent du summer school, ils savent tout faire pratiquement ce qu'on sait faire sur la transformation, le séquençage de génome, la cartographie QTL, ils font tout ça en quelques semaines. Donc, je suis pas content d'avoir Greg. Thanks so much for coming. It was a long introduction. I can tell you what I said when we are done.
Merci. Thank you. So thank you for the introduction, Kristen. Thank you for the invitation. It's really nice to be here. So as Kristen told you a little bit about my background, I'm a molecular biologist by training. I'm a yeast geneticist by trade, but I've always been interested in evolutionary biology and evolutionary questions. So my work really tries to span these two different disciplines of using yeast genetics and tools from molecular genetics to study fundamental questions in evolutionary biology. So at some basic level, my lab is interested in something that a lot of functional genomics is interested in, is the relationship between genotype. So in yeast, this is the 12 megabases of DNA organized along 16 chromosomes. How does phenotype encode genotype? And so in yeast, we talk about how does genotype encode phenotype. In yeast, it's often simple things like growth rate, forming colonies, ability to form a colony on a plate. And while a lot of functional genomics tools focus on modifying the genotype and looking how those perturbations change the phenotype, in my lab, we kind of do what's an older school method of we impose evolutionary pressures, we impose selection on phenotype, and we ask, how does the underlying genotype of the organism change in response to whatever selective pressure we put them under? And so part of the motivation from this work comes from people that study evolution in natural populations. So for a long time, it's been known if you have independent populations that are identical and evolving under similar conditions, that they'll tend to evolve similar evolutionary solutions to those selective pressures. So just to show a classic example of evolution in natural populations, I like to use the example of these cavefish. So we have a faculty member in my department now who studies the evolution of these cavefish species. And so they exist in two forms. You can find them across the US Southwest and in parts of Mexico. You have this normal freshwater fish. This will occasionally find itself trapped in cave environments. And when it does, it will adapt. And on the right are two different populations that adapted independently to the same environment. And you can see that they did so through similar morphological changes, loss of pigmentation and loss of eyesight. And what's maybe more interesting is that this parallelism even extends to the genetic level. So both of these populations on the right have different mutations in the same gene underlying loss of pigmentation. And so one of the questions that really drives the work that we do in my lab and motivates is, how surprising should this be that you get parallel adaptation even at the genetic level? How reproducible do we expect evolution to be? Now, in natural populations, you're sort of limited by what you can find. Sometimes you get these nice natural replicates, but what we want to do is do this in a really controlled laboratory environment. And so the technique that my lab uses is experimental evolution. So what is experimental evolution? You can imagine doing the following experiment. You can come into the lab one day, you can inoculate whatever bacteria or yeast into a test tube. You can then let that grow till saturation. You come in the next day, dilute these back into fresh medium, let it grow again till saturation, and just reiterate this process over and over again. And at the end of the day, you can then do an analysis and ask, how has our population changed in response to whatever this medium was, whatever this selective pressure is that we put them under? And this is essentially what we do in my lab, except that we don't use test tubes. We do all this in 96 well plates. We don't actually do all the dilutions by hand. So we use robotics to automate this process. So my lab has a liquid handling robot that we come in and set up every day. You can see it doing these serial transfers. And so grad students will come in, they'll set the deck up, they'll hit start on the robot, and it'll take all of their populations and do these dilutions for them. And so this combination of miniaturization down to 96 well plates, where we can have 100 replicates in a very small footprint, and automation allows us to handle hundreds or thousands of identical populations and evolve them in exactly the same environment. Yeast grows so we do this with yeast. Yeast grows very quickly, so it has a lot of the positive attributes you would have with using other microorganisms like E. coli. We can get 10 generations of growth per day. And so very quickly, we can get hundreds or thousands of initially identical populations and evolve them for thousands of generations under the exact same conditions. This technique of experimental evolution has other benefits as well. So along the way, we will periodically freeze down all of our populations. So we have our frozen fossil record that we can go back to. And so if we find something interesting in one of the populations that evolved, we can go back 1,000 generations and ask, does that trait exist in an earlier time? We can sequence the genomes from earlier time points, or we can even, and we have restarted the evolution experiment. If we see some interesting case where evolution veers off in a different direction, we can rewind the tape of life and let evolution of that exact population play out again in more replicates. I'll talk more about it, but we do a lot of flow cytometry to measure fitness. So in other words, how much better is an evolved strain than from where it started? And so we can take our evolved populations and we can mix them 50-50 with the fluorescently labeled version of the ancestor, the starting strain, and we can directly compete them in co-culture. And so this is sort of like a time machine experiment where you're taking something and you're moving it back in time, 1,000 or 2,000 or 5,000 generations, and competing it against something in its past. But of course, for much of what we do now, the most powerful tool in our arsenal is whole genome sequencing. And so sequencing costs have come down enough. It costs, if you do the calculations, about $1 to get 1x coverage of the yeast genome, that we can use whole genome sequencing as a readout of our experiments. So after we do several thousand generations of evolution, we can sequence single clone from each population, identify exactly what changes it has in the genome. We can do whole population sequencing, and we've done some time course sequencing. And so, like I said, we do this in my lab using yeast, and I'll get to some other benefits that we have of using this model system. But to give you an idea of some of the things that we can see about the evolution of these populations. So I'll show one population. This was one single well of one of our evolution experiments that we went back and we sequenced the entire population at multiple time points from our fossil record. And so this is a 1,000 generation evolution experiment. And on the y-axis is the frequency of all mutations that we can see within this population at a given time. And so you can see that we have a very clear window onto all the mutations that are arising and going extinct within this population. So this was an asexual population. And so early on, you had 2 mutations that arose and fixed within this population. Then you had this group of several mutations that came up, but because it's an asexual population, these got outcompeted by more fit mutations that came around later, and then these initial mutations went extinct. And so we can very clearly see the dynamics of adaptation within these populations. And here's just one other example that illustrates this clonal interference competition between different mutations that arise from the same background, because you had this mutation in blue that was increasing in frequency, but it got outcompeted by this group of mutations in black, and then everything got outcompeted by this group of mutations in purple. And so using experimental evolution and whole genome sequencing, we can get a really detailed picture of all the mutations that are coming up within evolving populations. But what a picture like this doesn't tell you is what are the mutations that are actually driving adaptation in these populations. So what are we actually selecting for within an evolution experiment? So if we take one of our evolution experiments, we sequenced about 40 populations. And from those 40 populations, we identified 723 mutations across replicate populations. And these 723 coding sequence mutations, if you were just to randomly throw this across the yeast genome, which contains about 5,800 genes, what you would expect is that most of those would have hit a different target. But what we actually see in our observed data is that we have a very small number of genes, 24 genes, that were mutated in at least 3 independent populations. And so this is a way for us to identify what genes are actually being selected for. And so just to zoom in and show what these 24 mutations were. And so this is a list from a single evolution experiment we did, where we sequenced multiple replicate populations. We had 21 different populations that had a mutation in the gene IRA1. And these are all the genes that are overrepresented in this experiment. And so these are likely the targets of selection in our evolution experiments. And we can actually look at what these mutations themselves are, and we find that they're almost exclusively nonsense, missense, or frame. For IRA1, if we look at it, we can see a mix of missense. Sorry. Thank you. That was a very quick technical change. So just as a reminder, we did a single evolution experiment with haploid yeast growing asexually in enriched glucose media. And then we sequenced 40 of those populations from which we could identify mutations that were overrepresented, identified multiple times across replicate lineages. And this is that list of genes. These are likely targets of selection in our evolution experiment. If we look then what mutations we're actually seeing, for IRA1, our top hit, we see a mix of missense mutations, nonsense mutations, and frame shift mutations. What this indicates to us is that when we get mutations in IRA1, we're selecting for loss of function because we see all classes of possible mutations. In contrast, for the things involved in cell wall assembly or cell wall biogenesis, we only see missense mutations for KRE6 and GAS1. And this suggests that this function, cell wall assembly, we're selecting for alteration or attenuation of function, not loss of function. And so I've shown you so far that using experimental evolution, we can identify, we can observe the dynamics of adaptation within populations. We can identify what genes and pathways are being selected for and how selection is acting on those. Here we can also take advantage of the fact that yeast are sexual, and we can use that to do genetics to actually identify, verify that these truly are targets of selection and to quantify the fitness effect of these individual mutations. So here's one example of a cartoon evolved population that has picked up 5 different mutations. And let's say we want to know which of these mutations, we can see them by sequencing, but we want to actually quantify the fitness effects of these mutations. We can take our evolved strain and we can back cross it to the ancestor. We can then let it, so we'll not make a diploid that's heterozygous for all of these evolved mutations. We can then put it through meiosis and collect a really large pool, hundreds or tens of thousands of these, that all have random combinations of these evolved mutations. We can then take this pool of segregants from this cross, and we can then just propagate it over time under the exact same conditions we did for the evolution experiment. And if we were to do that, we put all these in one tube and we would sequence these genomes. We would see all of these mutations would look heterozygous in this pool. But if we, like this, then if we impose selection on this, just propagate it in exactly the same conditions as the evolution experiment, we would see that these mutations would change in frequency, and they would change in frequency in proportion to their fitness advantage. So this provides us a really nice benefit where we can actually use yeast genetics and allow Mendel to shuffle the deck, and we can then quantify the fitness effects of every mutation within these populations. And so I'm going to give you 2 examples of this. So the first is a very simple population. This is one that was a 1,000 generation evolved population. And by generation 1,000, 3 mutations had fixed: ELO1, GAS1, and sterol12. If we backcross this, make a pool of segregants and propagate that forward over time, we can look at the change in these evolved alleles within this pooled experiment. We can see that ELO1 stays at 50%. So this is a neutral mutation that we've now separated from the beneficial mutations. And we can now quantify the fitness effects of sterol12 and GAS1 by just measuring the slope in the change of the log ratio of these 2 of them. Here's a more complicated experiment or population. By generation 1,000, I think about 23 different mutations have fixed. If we want to figure out which ones of these are actually driving this evolution experiment, we can do the same thing. We can cross this back to the ancestor, get a pool where we've shuffled the deck and we have all possible combinations, and propagate that pool forward over time. And when we do that, we see that most of these mutations, most of these 23 mutations are neutral. And if I highlight just those that have a positive slope, we can see there are only 3 beneficial mutations in this population: ROT2, IRA1, and MID2. And we can map these back to the dynamics of adaptation. And we can see that there were these 3 major selective sweeps that happened within this population. Each one had a single beneficial mutation and a bunch of neutral mutations that got pulled along for the ride. And so through this introduction, I was hoping to illustrate the power that we have with experimental evolution, that we can do many independent replicates of an experiment. We can then use sequencing to identify the dynamics of adaptation, the drivers of adaptation, and we can quantify the fitness effects of individual mutations. And this has long been the bread and butter of what we do in my lab. I want to tell two stories that illustrate the use of this power and bring up, at least in the one case, a very surprising observation. So I mentioned earlier that we often quantify fitness of our evolved populations by doing a competitive co-culture assay. And so a little more detail on how this works. We could go to the freezer and we can pull out some plate that has 100 populations that are frozen from some time point, and we can just do a competition experiment in the following way. We mix this 50-50 with a plate that has the ancestor strain with GFP being expressed. We then compete them in co-culture over time, and we look in the change of the fluorescent reporters. And so as the evolved populations outcompete the ancestor, you see a loss of the GFP population. We can very precisely quantify in a high throughput way fitness. And this is very accurate because our error is usually about 0.5% in fitness. And so this is a real data for multiple replicates of the same population. And so this was something we would do routinely. And in the process of doing some of these routine assays, we made an interesting observation about one of our populations. Again, this is a haploid population, evolved asexually in just standard laboratory medium, so nothing fancy. This population, the BYS1D8 is just our designation for it, evolved for 1,000 generation. There were 4 main selective sweeps. There were 3 mutations affixed here, a single mutation, and then 2 groups of mutations later on. From this population, we isolated 3 clones: the early clone from Gen 0, an intermediate clone from generation 335, right after this first selective sweep, and a clone from the endpoint generation we call the late clone. And in doing fitness assays, we observed that there was a gain of about 3.8% in fitness between the early and the intermediate. So the intermediate can outcompete the early at a rate of 3.8%. The late clone then can outcompete the intermediate clone at a rate of 1.2%. So if we do a co-culture experiment of the late and the intermediate, the late will win with a slope of 1.2%. And so the standard interpretation would be the late clone should then be 5% more fit than the early clone. However, when we did this experiment, competing the late clone versus the early clone in co-culture, we found a surprising effect in that the early clone could outcompete the late clone. So here you have a population where you had the ancestor strain A was outcompeted by B, C then outcompeted B, but C turns out to be less fit than where it started 1,000 generations ago. And so this was initially a very puzzling observation. And so we went back and did a lot more fitness assays to try to verify that this was the case. And we did something sort of on a whim, where we did the fitness assays, but not mixing things 50-50. We mixed them at all different ratios. And so we would mix the late clone from generation 1,000 and the early clone from generation 0, everywhere from 90 to 5 to 95 to 5 to 5 to 95. And when we do this experiment, we find that with the starting frequency of the late clone close to 0, the early clone wins, and this is what I showed before, around 50-50. However, if we start the late clone almost near fixation at 90-10 or 95-5, the late clone can actually win this competition. So this is a phenomenon that is known as positive frequency dependent selection, where the most abundant genotype has the highest fitness. There are very few things that show this attribute of positive frequency dependent selection. And one of the ones that is most often seen are these toxin antitoxin modules. And yeast really only have one well-characterized toxin antitoxin system, and that's the yeast killer virus. This was something that was known and was thought to be absent from the strain we were using. However, we later confirmed that our strain, in fact, does have this. So what is the yeast killer virus? What is this toxin antitoxin system? So yeast can contain cytoplasmic encapsulated double-stranded RNA viruses. And so here's the killer virus. It's a double-stranded RNA, and it encodes one and only one open reading frame. And that open reading frame encodes a toxin that gets secreted and kills non-killer containing yeast. The pre-processed form of this toxin confers immunity to the toxin itself. So that's how killer containing yeast don't kill themselves. There's a very easy assay to determine whether or not your strains contain killer toxin. And you can plate a lawn of a sensitive strain, which we've done here. This is the lawn. This just has a blue indicator dye in it. And then we put 2 dense spots of either a killer containing or a killer absent strain. And you can see a zone of clearing around the killer positive, where it's killing and it's producing a zone of clearing in the sensitive lawn. So using this assay, we were able to confirm that our strain does in fact have the killer virus. But because killer virus has one and only one open reading frame, it can't actually make copies of itself. And so the killer virus requires another virus, the helper virus. This is really the true virus, the Todi virus. It's another double-stranded RNA encapsulated cytoplasmic virus. It encodes 2 open reading frames. One is the capsid protein, and one is a capsid polymerase that's made from a translational read-through. So all helper can do is make more copies of itself. And then killer is the satellite virus. It's really a selfish parasite of the helper virus, because it can steal some of the polymerase and capsid from helper to complete its life cycle. And so we then confirmed by RT-PCR that the strains we were using that, according to the literature, did not have these viruses, do in fact have the M1 killer virus and the K1 killer virus and the M1 helper virus. And so, unbeknownst to us, we've, in my lab, have not just been doing yeast experimental evolution, we've really been doing yeast virus coevolution. And so we had freezers full of these populations that we've been evolving and focusing on the yeast genome, but we've been ignoring the fact that there's been another subgenome there this whole time. And so once we learned that, we had to go back and to actually look at what's happening to the killer virus in some of our evolving populations. So using the same ones I was showing you before, haploid yeast evolved asexually in rich glucose media. We went back to the freezer. We pulled out from our frozen fossil record a whole bunch of time points, and we then looked at killer virus. So just to start, we picked out about 150 populations, and we came up with a quantitative scoring metric for killer activity, where 1 was a large zone of clearing, meaning it's the ancestral phenotype, it has a fully functional killer virus, down to 0, where there's no killing ability detected. And for the 142 different populations, we looked at different time points, and we can see that over time, we did get an increase in the number of populations that had some loss of killing ability. We then set up the inverse assay to ask about the other phenotype, which is immunity or resistance to the toxin. And so to do this, we again use the robot to do a high throughput assay, where we put a diffuse spot of our query strain or evolved strain, and in the middle, we put a really dense spot of a known killer containing yeast. And then we again had a phenotype where the ancestor would have no halo because it's resistant to the killer toxin, down to ones that are now fully sensitive. So I'm going to show this data now on 2 axes. So on the y-axis here is killing ability. So again, you can see about a quarter of our populations had some loss in killing ability. And of those that lost killing ability, about half of those then developed some sensitivity to the toxin. And just as a way of sort of illustrating the richness of the data we have, we can look in at the ones that lost both killing ability and develop some sensitivity to the toxin, we can look as a time course. You can see for most populations, you had the loss of killing happen quite early, followed by a much later loss of resistance to it. But there were some populations where we have, trying to find a good example, where we have almost simultaneous loss of both killing ability and immunity to the virus. So to try to test whether or not this non-transitivity, this B beats A, C beats B, but then A beats C, if this non-transitivity and this frequency dependence were attributable to the presence of this killer virus, we went back to this experiment of competing the early and late, and we removed the killer virus from the situation because we can cure the virus from these populations. And when we do that, we found the expected result that we were seeking initially, that the evolved population, the endpoint of 1,000 generations, really is about 5% more fit than the ancestor. And so how did this non-transitivity arise? Because here we have a case where it's a direct line of evolutionary descent that you ended up at the end being less fit than where you started at the beginning. Well, we went back and we started sequencing these viral populations to see what we could identify. Using RT-PCR, we sequenced all of the viral populations where we saw a change in phenotype. So this is the killer toxin genome. This is the double-stranded RNA. This is the one open reading frame that gets processed. There's a polyadenylation site, and then you have these hairpins that are required for replication and packaging of this virus. When we sequenced the genome, we found mutation. When we sequenced the entire virus, we found mutations only within the coding sequence, and we found a huge enrichment for missense, nonsense, and frame shift. So it turns out actually that the viral genome was the largest target of selection overall in our evolution experiments. And we found a very good correlation between those that had a change in killing ability in the presence of mutations. In doing this sequencing, we actually observed some really surprising things, that we found some viruses that seem to have an odd structure. So these are two examples of viruses that we could sequence where we lost either most or essentially all of the open reading frame. And we found viral particles that have become so small that they basically retain nothing but the information required for packaging and replication. And so these are what are known in the field as defective interfering particles, that they get in and they can just make copies of themselves and shut down the system. And it turns out that this is what was happening in our population where we observed non-transitivity. And so to try to summarize what we found from our evolution of non-transitivity experiment, here again is that population. It evolved for 1,000 generations. These are all the mutations in the yeast nuclear genome and their dynamics over time. We started off with a population, the early clone, that could produce and was resistant to this toxin. So here it is. It has full killing ability and it's fully resistant. It then got outcompeted by an intermediate clone that picked up 3 nuclear mutations, including one that we identified as UR1 as a beneficial mutation. This led to the more fit nuclear genome. And at the same time, there were mutations within the intracellular viral population that caused loss of toxin production, though they retained immunity to the toxin. Then when we got to the late clone, they fixed an additional 10 mutations, including a known beneficial mutation in the gene sterol2. So again, the yeast nuclear genome got more fit. And there was additional mutations within the viral population, making these small interfering particles that now not only were they not able to produce toxin, they were now sensitive once again to the toxin itself. And this was our late strain, such that if we then competed our late clone against the early clone, we would see a non-transitive interaction where the late clone would actually lose to the early clone, because it's producing toxin, and that loss would be show positive frequency dependent selection. And so this was a real surprising observation to us because it suggests that evolutionary lineages, so direct lines of evolutionary descent, don't always need to be transitive. And so we like to think of evolution often as these non-transitive interactions where you have rock, paper, scissors effects happening over evolutionary timescales. And it's also interesting to think about in the context of fitness landscapes, where we talk about, peaks and valleys to a fitness landscape. But here we have a population that at every single step it took along the way, it was increasing fitness. And so this was always moving uphill, yet at the end of the day, it ended up less fit than it was where it started. And so it suggests that in some ways, there could be these weird discontinuities in a fitness landscape. And you might think of fitness landscapes more like these Penrose staircases as opposed to a true landscape. And so this was one way of looking at using experimental evolution to look at interactions between two genomes, the yeast nuclear genome and a cytoplasmic viral genome. And all that I've talked about, I think I've said the reminder many times, has been the simplest, dumbest experiment we can do. Just haploid yeast growing asexually in rich glucose, standard laboratory media at 30 degrees. But what I want to point out is that experimental evolution is a really powerful and versatile system. We have all these different knobs that we can turn. And my lab is currently, and has been for the last 10 years, trying to turn as many of these knobs as we can. The one thing we can do is we can change the growth media. We've done evolution experiments. We grow them in different environments, fermentable and non-fermentable carbon sources. We will change, they'll alternate between fermentation and respiration. We can do them in environments where we turn the incubator up 1 degree every 10 generations or 100 generations and ask if evolution can keep up. We can do a lot to change the environment. We can also change how the robot does solutions to investigate fundamental population genetic parameters, such as increasing the population size, introducing migration between different populations. But most of what we've been doing, especially lately, and then we always have all of these tools at our disposal. We maintain the frozen fossil record. We can do quantitative fitness assays throughout, and we can use next generation sequencing to see how things are changing. But most of what we've been really focusing on has been using this experimental evolution paradigm and putting different things into our procedure. And so we've been changing really what we inoculate with. And so we had a whole lot of papers right after I started my lab, especially at Lehigh, looking at the rate of adaptation in haploids versus diploids. And a lot of it is based upon the assumptions of our new beneficial mutations dominant or recessive. And so we have a whole bunch of papers looking at showing that diploids actually evolve more slowly, and that is because they don't have access to recessive beneficial mutations, that the mutations they do get tend to be at least partially dominant, if not over-dominant. Recently, we've been doing a bit of work trying to put rare human disease mutations into yeast. And it might seem weird because a lot of the diseases that we're looking at will have, in humans, they'll manifest as neurological defects or skeletal defects. But in reality, these mutations are in very highly conserved biological pathways like glycosylation, translation. And whenever you put these mutations into yeast, inevitably they just lead to the phenotype of slow growth, which we're really good at identifying. And so I like to say that, so my postdoctoral advisor, David Bussing, who Christian mentioned at the beginning, used to always say, a selection is worth 1,000 screens. And so experimental evolution can take what would be a screen looking for bigger colonies in a field of smaller colonies on a plate and turn it into an enrichment selection. And so we've really been scaling up, using CRISPR-Cas9 to make human disease mutations, and then evolving them to identify and enrich for suppressors that partially compensate for these. And we had positive experience with doing this once, so now I have a new grad student who's really scaling up to make a large panel of these. But what I want to talk about next, and this is really the first time I've talked about this, is the most recent thing we've started, which is looking at hybrids in yeast. And so again, much like the story I just told you, this is looking at the interaction of two different genomes. Rather than a nuclear and a intracellular viral genome, we're going to be making hybrids between two different species. So this is the phylogeny, the genus Saccharomyces, the one that we all know and love because of its use in beer and wine production is Saccharomyces cerevisiae, but there are 8 different recognized Saccharomyces species. Its closest sister species, Saccharomyces paradoxus, has about 87% amino acid identity. But this is still a lot of divergence. I think it's roughly supposed to be like human mouse level amino acid divergence, but yet these genomes are completely syntenic. They have the same genes in the same order, and they can form hybrids naturally. And so there are a lot of hybrids that we find associated usually with fermentation, with beer production. And so these are just a few examples. You even have hybrids between 3 different species. And so this would be the lager yeast that makes the lager beers. We decided to study hybridization using Saccharomyces cerevisiae, the well-studied laboratory model, and its most closest sister species, Saccharomyces paradoxus. So cerevisiae is typically associated with fermentation. Paradoxus is a wild species. Like I mentioned before, these genomes are syntenic, same genes, same order, same 16 chromosomes, and they will mate either in nature or in the lab. However, when they mate, they will form a diploid that has 1 copy of the cerevisiae genome, 1 copy of paradoxus. They can grow asexually. They can undergo mitosis just fine. Where they fail is in meiosis. If you try to ask them to undergo recombination and segregate their chromosomes. And this is where the species barriers lie within this genus. So just to show that, if we take Saccharomyces paradoxus, cross it to itself, we get a diploid. We can then sporulate that diploid. It will make 4 meiotic progeny. And in yeast, we can go in with a needle and we can separate those meiotic progeny, put them on a petri dish, and they will get something that looks like this, where these 4 spots are the 4 different meiotic progeny from a single diploid strain. And so here you can see almost all of these spores have germinated. Just about every meiosis was successful. And this is what you would see within a species. If we were to do this between cerevisiae and paradoxus, we would get a plate that looks pretty pathetic. Almost all of the tetrads failed to germinate. There's only 1 spore that germinated, and even this is actually probably the best plate that we would ever have made, because usually you get no viable spores out of this cross. So why is that? They have the same genes, the same order, they're syntenic. It's not rearrangements. The reason why yeast intercrosses in yeast fail is because of the sequence divergence between the chromosomes. And when the chromosomes line up, that there's mismatch repair that will reject pairings between heterologous chromosomes. What's nice about that is if there's a surveillance mechanism that's rejecting these interspecies crosses, we can just shut off that surveillance mechanism. And so we can take Saccharomyces cerevisiae and Saccharomyces paradoxus. We can put 2 genes involved in mismatch repair, MSH2, the main player in mismatch repair, and SGS1, which is a helicase. We can put them under the control of a promoter that is off in meiosis. So now what we're doing is when we make a diploid that's heterozygous between these, we shut off the surveillance mechanism that would be rejecting crossovers between heterologous chromosomes. And in doing that, we can get a plate that looks better. It still doesn't look good, but it looks better. And here we can actually see what we're looking for, a 4-spore viable tetrad. Where meiosis was successful, it made 4 progeny, each of which have a full complement of 16 chromosomes. And if we show what this machinery does, is we can increase the spore viability to about 25%, and we can get about 3% of the cases do we get a full 4-spore viable progeny. And we've actually found other tricks where we can try to fax sort ones that are likely to produce 4-spore viable. We can get this up to, to pretty close to wild type in doing that. But using this method, we isolated 20 F1 progeny between these 2 species. And so these are not diploid progeny. These are ones where we've actually had recombination, and half the genome is now coming from one species, half the genome from another. And we sequenced all of these. So this is these 20 F1 F1 hybrids that came from 5 different tetrads. You can see the proportion of their genome that comes from Paradoxus in red or Cerevisiae in blue. And we can also genotype where their mitochondria are coming from, if it's coming from Cerevisiae or from Paradoxus. Just to zoom in on one of these tetrads, tetrad 1, you can see that we really do have lots of recombination events. Here's another example, another one of the crosses. So a few things to point out is that we always have uniparental mitochondria. So there's not mitochondria from both parents present. And then we have an average of 17 crossovers per spore, which is good, but it's a lot less than the 70, 90 crossovers you would get for an interspecies cross. But nevertheless, we accomplished, and there are still, I want to point out, there are still some uniparental chromosomes that have come through. So normal yeast meiosis, you would not have uniparental chromosomes because of the 70 and 90 crossovers. And so we were able to identify, isolate, and genotype F1 progeny that are haploids that have half their genome from one species, half from the other. So once we had this, we want to ask if we can do basic mapping of traits. And so one trait that was really obvious was this clumpiness, that the Paradoxus strain would grow in these little clumps, whereas the Cerevisiae strain is a lab strain, is well behaved, and you usually see as single cells. And this clumpy trait segregated 2 to 2 in all of our crosses. And using only just these 20 segregants, we were able to map this clumpy phenotype to a region on chromosome 2 that had a gene that was well known to be associated with this trait, AMN2. And so we can show that even with 20 of these F1 hybrids, we can start mapping traits, at least Mendelian traits, and crosses between different species. However, most of the traits that we've been identifying are really quantitative. And so here's an example of an experiment where we just wanted to ask, at what temperature can our F1 hybrids grow? So on the left, these 2 boxes are the parents. Again, this is Cerevisiae, and this is Paradoxus. They can both grow over this full range of 16, 30, and 37 degrees. But if you look at the F1 hybrids, they almost all have reduced temperature tolerance, meaning that they usually are temperature sensitive at the high end and temperature sensitive at the low end. And our interpretation of this right now is their protein complexes, because the parents are able to go robustly over this range, it's not that they're individual proteins that are temperature sensitive, but it must be these forced heterologous protein complexes, where some of the subunits come from Paradoxus, some come from Cerevisiae, that are unable to function at higher or lower temperatures. And this is what we want to map. But I do have one slide to illustrate that we don't know much about genetic interactions between species in yeast. We do know a lot about genetic interactions between species that involve the mitochondria and the nucleus. And so here's one nice example of a nuclear mitochondria incompatibility between Cerevisiae and Paradoxus, where you have the Cerevisiae MRS1 gene, which is involved in splicing. MRS1 is involved in splicing introns out of the gene that encodes cytochrome oxidase, which is a mitochondrially encoded gene. And the Paradoxus COX1 has 2 introns, whereas the Cerevisiae mitochondria only has a single intron. And so the mitochondrial MRS1 is able to remove the single intron, but if you build a strain that has the Cerevisiae version of MRS1, but the Paradoxus version of COX2, the MRS1 from Cerevisiae is unable to splice out one of the introns, and you get a non-functional cytochrome oxidase, a non-functional electron transport chain, and you become respiratory deficient. And this is one example, but there's been a number of nuclear mitochondrial incompatibilities identified between different yeast species. But there's never been any strong nuclear incompatibilities between species identified. We're working on the hypothesis based upon the temperature data that I just showed, that even though there might not be any strong, what you would call Bates and Dubzhansky-Mueller nuclear incompatibilities, that there's probably a large number of weak negative interactions between these two nuclear genomes. And so the way we like to think of this is puzzle pieces. But here you might have a same species protein complex where these things fit really well together. They stabilize each other, and this protein complex can function just fine. If I had made the green one, it would be the same. It's just a heterologous species. But when we make these F1 haploids, we're forcing heterologous proteins to interact in a complex. And we think that these interactions are failing, and that's why we're getting loss of growth at high or low temperatures. And so again, we built these strains because this is a perfect tool for our platform of experimental evolution. And so we're now evolving these strains, expecting to find that after evolution, we'll be able to find mutations that restore the normal functionality of these protein complexes. And so how are we doing this? This is all very new data, but we took our 20 F1 hybrids. We then made them diploids by switching their mating type and mating them to themselves. So we also have a homozygous diploids, but there's still half the genome from each species. And we also made the true heterozygous diploids of just mating Cerevisiae and Paradoxus, where they now have the entirety of both subgenomes. In addition to doing the temperature tests, we measured fitness, so our typical competitive growth assay. And despite a few interesting cases where some of these F1 hybrids are more fit, most of them are a lot less fit than either of the parental strains. So we do see a loss of fitness that is consistent with our hypothesis that you're making a lot of these weak negative genetic interactions. We've evolved these now. This says, generation 3000. We're almost at generation 5000 at this point. And this is an animated slide like crazy, but we can see that when we check this at every about 400 generations, we see continual gain of fitness, such that now all of our F1 haploid hybrids are now about as fit as the evolved parental strains, and they've all recovered most of their normal fitness. If we look at the diploids, we see the same story. All of the F1 hybrid homozygous diploids have increased fitness to be now about equal to that of the parents. And you can see from this as well that the correlation between the fitness in the homozygous diploids and the haploids is quite consistent as well. And so we do find, different initial fitnesses, but that all of the evolved haploids and diploids have regained sort of their normal growth rate. So we just did a large sequencing experiment. We actually went back to generation 2000 to sequence. It just takes that long to analyze the sequencing data. The evolution experiment has now moved on a few thousand generations. And so we just got our first sort of picture now of what mutations are happening within these hybrid genomes. So this was sequencing of the diploid homozygous haploid strains. This is a heat map. So these are the top genes. These are all genes that we identified at least 3 mutations, I think, across replicate lines. So these are things that are likely enriched for the drivers of adaptation in our experiment. And what I'm showing in this panel are the parents. So this is just the Cerevisiae, Paradoxus, and then the Cerevisiae Paradoxus hybrid in the third. These are the 20 different diploid hybrids. And then these are the genes on the y-axis and their enrichment score. The darker, the more replicate populations we've seen with the mutation in that gene. And this bar graph over here shows whether or not those mutations are occurring within the Paradoxus or within the Cerevisiae gene. And there are some, such as this BSC1, that when we see mutations, they're almost always ones where it was the Paradoxus gene present in that strain that got mutated. But our top hit, HSP104, we see mutations equally in the HSP104 if it had the Cerevisiae copy or the Paradoxus copy. We can show just another way of plotting the data to show the significance. So we can calculate p-values for how significant this enrichment is. And these are the 5 most heavily enriched genes within this study so far. The only thing we can notice is that we see 2 of these, so the SIS1 and HSP104, interact with each other. They're both heat shock proteins. And so we actually think that there's, again, this is supporting the idea that there's some aspect of maintaining stable protein complexes that has gone awry in our F1 hybrids. So this is very new data. We're really just starting to understand it, but this is what we expect to be working on for the next few years, because we think it's going to take a large number of generations to actually see the restoration of these weak negative interactions. But just to summarize everything I talked about, so we're really motivated by this idea that in natural populations, you see reproducible patterns in evolution, that we can use that to identify what's happening in the genome in response to selection. We do this in the lab using the technique of experimental evolution. It's a very powerful technique that we can maintain hundreds of populations for thousands of generations. We can use sequencing to then identify all the mutations that are occurring within these populations and how they move over time. You get a good picture of the dynamics of adaptation. Because yeast are a eukaryote, we can do genetics with them, and we can then identify beneficial mutations and quantify the fitness effects of those. I then told you 2 stories, both about interactions between different subgenomes. One was this yeast killer virus. We identified that our yeast strains, unbeknownst to us, had a second genome, this double-stranded RNA intracellular virus, and that the presence of this virus and the coevolution between the host and the viral genome led to this interesting observation of non-transitivity along a direct line of evolutionary descent. And at the very end, I talked about our most recent work constructing interspecies F1 hybrids, where the genome is now forced to have half of its component from one species, half from another. And then our use of experimental evolution, we're hoping now, as we analyze this data, to identify mutations that are compensating for weak interactions between heterologous proteins in complexes. So with that, I should just thank the folks in my lab who did this work. A few people have left that contributed to some of this stuff, as well as our funding. And thank you for listening.